Students, in this video lecture, we will see the use of transgenic animals, ethical issues, and biopiracy. So first we will see what are the transgenic animals and how we are using these transgenic animals and what is the utilization of these animals. So transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. It means an animal is having foreign gene to produce some foreign protein material is known as transgenic animal. The transgenesis is the process by which mixing up of a gene takes place, and transgenic technology has led to the development of fishes, livestock, and other animals with the altered genetic profiles which are useful to the mankind. Until date, rat, rabbit, pigs, sheep, cow, fishes have been produced, and a total of 95% of the transgenic animals have been made alone by utilizing mice only. Now we will see what is the use of these transgenic animals. So first point is to study the physiological and developmental process in the animal. Means these transgenic animals have been utilized to study the normal physiology and developmental process. So to study the regulation of a gene means how it is regulated, and to assess the functional role of that particular gene in the physiology and metabolism, the transgenic animals have been produced. So by manipulation in a particular gene, we can study the effect of altered functional role of this gene, and the alteration could be, it means we can alter the expression of a gene by the overexpression. Means in this kind of experiment we use to increase the protein production of the animal. Means a particular protein will be overexpressed. And second kind of alteration we used to do is knockdown. In this kind of experiment, we used to decrease the protein production of a particular gene. And third is knockout experiment. In this experiment, we used to delete the a gene so that the protein will also be deleted. Means by this kind of alterations, mostly we utilize to produce several transgenic animals. Suppose we want to study a particular gene's function, then either we will overexpress it, either we will knock down or knock out it, then we will see what is the effect of the alteration of that particular gene in that animal. Second is the study of disease. So the transgenic animals are designed in such a way as we can understand how a particular gene uh, contribute for the development of a disease. The transgenic animal of the human disease model can be utilized in investigation of different treatment methods also. For example, the animal model of cancer, cystic fibrosis, rheumatoid arthritis, etc. have been already been studied by utilizing the animal model of the human disease. Third is for the biological product production. So the transgenic animals can produce a particular protein for the treatment of several human diseases. These animals have been created by introduction of a particular gene or a DNA fragment which codes for a particular protein. For example, alpha 1 antitrypsin to treat emphysema. So we can utilize a fragment of gene which encodes this particular protein, and we will insert in a vector, and we will introduce this vector in the animal, so that that animal will start producing this particular protein. The first transgenic cow was was produced in 1997, and that cow have been producing the milk with enriched alpha lactalbumin as to provide a balanced diet for the human babies. It means we can utilize these technologies to produce several products which can be utilized for the human welfare. Fourth is the vaccine safety. So these transgenic animals have also been utilized as a human model for the vaccine testing. For example, poliovirus. It means before you before injecting a particular vaccine in the human, we can uh, we can do a trial in the human model for the vaccine assessment. Means if there would be any adverse effect due to vaccine, then we can assess in that particular animal which we are using as a human model. Fifth is the chemical safety testing. So the toxicity or safety testing is also being done for the drug testing before starting in the human trial. And in this case, the transgenic animals are made by a manipulation of the gene to make more sensitive animals towards the cytotoxicity, cytotoxicity of the drug. It means to assess the cytotoxicity or safety testing, we use several transgenic animals with the manipulation of several genes, so that we can assess that either this animal is affected or not. Now we will see what are the ethical issues in producing these transgenic animals. Since the testing of drugs, chemicals, vaccines, and various products of the animal is mostly harmful to that particular animal. So some ethical standards are required to evaluate the morality. So the genetic modifications of of the organism can have unpredictable results, and it may harm the ecosystem too. It means since we are using different genes to produce different products in animal models, so it is ultimately harmful to that animal, because we are doing the experiment and we don't know what is the fate of that particular protein. So ethically, uh, it is not good. So, as per the morality basis, there are some guidelines we need to follow for the production of genetically modified organisms. So, there is a government body, GEAC, Genetic Engineering Approval Committee, and that organization make decisions on GM research, means genetic modification research, and safety of their products in the animal as well as the human being also. And there is an important issue in granting patent also. So we will see what kind of problems we could face with the transgenic animals. So there is a very this is a very debatable topic, as we can provide a patent to the newly prepared transgenic animal or not. So there is an important issue in granting patent, just because it is a debatable topic to provide a grant in GM organism. Some people are against the patent of GMOs because they used to utilize the of product or technologies by which the naturally occurring genes. It means people used to use the naturally occurring gene and they used to produce GMOs. So it is very difficult, either we should provide the patent to that animal or not. So as we know that rice is an important crop in Asia and basmati rice is a breed which have been cultivated in India for a long time and several evidences, documents are there which reveal its origin that they have been originated from India only. But in 1997, an American company got patent and trademark for the basmati rice. So several attempts have also been made to receive the patents of Indian traditional herbal medicines, turmeric and neem, etc. So people used to exploit the traditional medicines, traditional processes of the another country. So there is a problem of granting patent on such organisms, including GMOs also. So now we will see what is the biopiracy. So use of bioresources by multinational companies and other organizations without proper authorization from their native countries. And most of the financially rich countries, those are developed countries, do not have a rich biodiversity and traditional knowledge. However, on the other hand, the developing and underdeveloping countries are rich in both terms of the bioresources. Means the developed country, some most of the developed country, they do not have the rich biodiversity and traditional knowledge. And the other hand, the developing or underdeveloping countries, they used to have a rich biodiversity and traditional knowledge. But the advancement of the technologies in the biological system, the the uh, developed country, they utilize the knowledge, traditional knowledge and biodiversity of the developing and underdeveloping country, and they used to get the patent. So the say though they utilize, or we can say they used to exploit the knowledge and the bioresource of the developing and underdeveloping uh, countries. But there are some laws which protect, or we can say they provide a favor to the native or the biological products, and they the laws mostly support the developing and underdeveloping countries. Thank you.